Okay, this is round 2. Hopefully it works. Last time my sound wasn't working. So, describe the so the contractile filaments in a skeletal muscle. These are slightly different than smooth muscle. So, I'm gonna try to make that actually parallel. So in skeletal muscle, there is a protein scaffolding. It's like the framing of a house, basically, made out of Z discs, which are these parallel lines right here. Nebulin, which would be this right here. And titin, which is this. And this protein scaffolding is what allows for the binding of, it just is kind of going to hold the contractile filaments. So we kind of have to know the anatomy of this to understand the contractile filaments. Now, the contractile filaments, there are 2 main contractile filaments, actin and myosin, but actin has lots of parts. So I'm going to talk about actin first. So actin, there's G-actin. And G-actin is just a special kind of actin that has binding sites for ATP. So ATP can come in and bind in these little circles right here. And G-actin actually strings together in a special way in an alpha helix. So it looks kind of like, my drawing is bad right now, but it strings together kind of like this with the ATP binding sites out. I lost my questions. There we go. Like that, along like a central line in an alpha helix shape, similar to the shape of DNA as an alpha helix. And this, once we're in an alpha helix, is called F-actin. And F-actin stands for fibrous actin because it's kind of in a long fiber. G-actin stands for globular actin. F-actin, in F-actin, these ATP binding sites are no longer ATP binding sites. They are called active sites. And they are where myosin is going to bind. So when myosin binds, it binds to what was the ATP binding sites on G-actin that have now been through a conformational shift that make them myosin binding sites. And this F-actin winds around nebulin. So it's nice and tightly alpha helix coiled around nebulin. I'm drawing that as best I can, but I'm not an artist, so work with me. It'll make sense, I promise. Hopefully. So we have the alpha helix around nebulin, and that's part of actin. Then, also part of actin is, we call it tropomyosin. Now this is going to cover up F-actin. So it covers up F-actin, the binding sites for myosin. So it covers the myosin binding sites. That's G. And for every 7 G-actin, there's 1 tropomyosin. But for the sake of my drawing, I'm just gonna draw here some tropomyosin, if I can make it work. So tropomyosin is gonna cover up the myosin binding sites on the F-actin. Just like that. And really, it should probably not have any spaces in it. And then the third component of actin, the last component of actin, is called troponin. And troponin is a 3-component complex. There's I, T, and C troponin. I troponin has a high affinity for F-actin. T troponin has a high affinity for tropomyosin. And C-troponin has 4 calcium binding sites. So it has 4 calcium binding sites. And these calcium binding sites, there are 2 that are high affinity and 2 that are low affinity. So when there's a high concentration of calcium in the cell, these fill. So this is high concentration filling. And this one, these are filled all the time. So they're filled even at low. Oh my gosh, I'm making a mess of this paper. They're filled even at low concentrations because they're high affinity. So the calcium will bind even if there's not very much calcium. This part of troponin is what keeps the troponin complex together. So when people have heart attacks, they test for troponin C to see how bad the damage is, pretty much. And the troponin actually binds to tropomyosin. So there's 1 troponin for each tropomyosin. So I'll try to show that, but I got a little crazy with my tropomyosin drawing. So for each tropomyosin, there's 1 troponin molecule or complex. So I'm just going to kind of draw here, but so it's a 1-to-1 ratio, and it binds to tropomyosin. So I'm just going to say that 1-to-1 ratio. And the role of troponin is to, when calcium binds, troponin moves tropomyosin off the active site of F-actin. And then I keep saying myosin and all this. So myosin is the other contractile filament in skeletal muscle. So myosin, there's actin and then there's myosin. And myosin is made up of 2 main parts. So that has a heavy chain, and the heavy chain has a tail, which is this long part right here, a hinge region, which is like this part right here, where the tail obviously connects to what we call the head, and then it has a head. And each myosin molecule has 2 heads. So there's a hinge region where the head can move and kind of switch angles. And then each myosin also has light chains, and it has 2 light chains, 1 alkyl light chain and 1, it's leaving me, but hopefully it comes. Regulatory. I'm like thinking really hard. Okay, and 1 regulatory light chain. So that's myosin. Myosin also has 2 different conformations. So it can move freely between 2 different conformations. We call this one cocked and this one uncocked. And that becomes important when we actually work with contraction. But myosin binds to titin, so each titin will have a bunch of myosin on it. This isn't necessarily the most accurate drawing of what things will look like, but that's definitely not the right way. There's a bunch of myosin there. And when they go through a conformational shift, it'll make the muscle cell smaller. The sarcomere, which this represents 1 sarcomere right here. This is a sarcomere, which is just like a unit in the muscle cell. And we talk about sarcomeres because that's where contraction happens. Contraction happens in the individual sarcomere. And sarco just is a prefix for muscle. So when we say sarco or myo, then we're usually talking about muscles. Actually, we are talking about muscles whenever we say that. So knowing that, we can kind of talk about how muscles contract. But we also have to talk about how an action potential actually reaches the cell where, like the sarcomere, where things can happen. So if this is a neuron, then we have voltage-gated calcium channels here, and calcium is going to flow into the cell when an action potential travels down. So we have an action potential travel down, calcium is going to flow into the cell, and we're going to release neurotransmitter because we have endocytosis that is induced by calcium. So we're going to release neurotransmitter. Those neurotransmitters are going to bind to the muscle cell and have an action. They're going to make an action potential happen. So the action potential will travel along the muscle cell as well, opening voltage-gated sodium channels along the way. So we are depolarizing the membrane. But muscle cells are really, really big, so they have lots of space, and it's not enough to bring enough calcium into, we open voltage-gated calcium channels here too. It's not enough to bring calcium actually into the part of the muscle where we have contraction into the sarcomere. So muscles anatomically have these things that look like if you had a potato and you stabbed holes in it so it doesn't explode in the microwave. It's like those indentations that make it so the action potential can run through and get closer to the inside of the cell so that we can actually get close or closer to where the sarcomeres actually are. And these are called T-tubules. And the T-tubules are where the magic starts to happen. So T-tubules so I lied about this earlier, this whole voltage-gated calcium channel thing. The T-tubules are the first place you're going to see a voltage-gated calcium channel. So this is a voltage-gated calcium channel. T-tubules also have voltage-gated sodium channels, which are open with an action potential. They have voltage-gated potassium channels. They also have voltage-gated chloride channels, which I'm put on this side just because I don't have enough space, but they could go on either side really. They also have voltage-gated chloride channels, which chloride is a really big deal because potassium, when it flows out of the cell, as it does because that's where its driving force wants it to go, it actually builds up positive charge outside the T-tubule and causes a problem, so our resting membrane potential isn't at the right spot anymore, and you would get a lot of action potentials that you don't want if we didn't cancel out the positive charge with negative charge. So we have chloride that also flows into the T-tubule when the voltage is right. So if there's too much potassium, we let some chloride out. There's also chloride leak channels to help cancel that out. So about 80% of resting membrane potential is because of chloride. And that's only in T-tubules. That's not everywhere, but that's in T-tubules. Anyways, that was a random tangent. So then, right next to the T-tubule, we have something called the sarcoplasmic reticulum. And the sarcoplasmic reticulum is basically the muscle version of an endoplasmic reticulum. In fact, it is the endoplasmic reticulum of a muscle cell. We just call it the sarcoplasmic reticulum so that we know we're talking about a muscle. And it has another channel that actually connects to the voltage-gated calcium channel. And it's called the ryanodine channel. So this is the RYR, the ryanodine channel. Ryanodine channel, and it's a mechanically gated channel, so it's not voltage gated. It does not open because of a depolarization of a membrane or because of different ion concentrations. It actually opens when the voltage-gated calcium channel opens. So if the voltage-gated calcium channell opens, the ryanodine channel is also going to open because it's mechanically gated. So in a skeletal muscle cell, the voltage-gated calcium channels literally do nothing except for open the ryanodine channel. They have no purpose because our calcium comes from only intracellular sources, which is different from smooth muscle. Smooth muscle calcium can come from intracellular or extracellular sources. So voltage-gated calcium channels are more important because we can get calcium flowing inside the cell from outside the cell. But in the sarcoplasmic reticulum, we have calcium that is being held here by proteins. I can never remember the name, but there's a protein holding them there so that they don't diffuse throughout the sarcoplasmic reticulum, because even though in my drawing it's pretty small, in a real cell it'd be really big and they would diffuse throughout, and there wouldn't be really a high enough concentration for a lot of diffusion to happen, and it might not happen fast enough for our muscle cell to contract. So when the voltage-gated calcium channel opens, calcium is going to flow out and actually down into the sarcoplasm, which is just the cytoplasm of the muscle cell. Sorry, I had to make sure my mic was on. I just got worried. Anyways, so calcium's going to flow down, and then that will allow it to get to the troponin. It's going to bind to troponin, and contraction is going to happen. And the mechanism for contraction I'm going to explain in a second, but I'm just going to finish up this really fast. So then, after our calcium flows out, we have pumps along the sarcoplasmic reticulum. They're called Sirca pumps. And they basically are just specialized calcium pumps. They pump calcium using ATP. So they are ATP pumps for calcium. So we just pump calcium back into the sarcoplasmic reticulum using the Sirca pumps. And they pump calcium really fast. So calcium is pumped back into the sarcoplasmic reticulum almost as fast as it flows out of the cell. Obviously not quite as fast because it's being pumped against its gradient. So we can't do it as fast as we can let it out of a cell. But there will be some calcium that diffuses and binds to troponin, but lots of calcium will also go back into the sarcoplasmic reticulum so that we don't have uncontrollable muscle contraction or too much muscle contraction that we didn't want. Once that calcium leaves, though, we have binding to troponin, and I'm going to draw, we call it the cross-bridge cycle. So myosin starts unbound with a phosphate and an ADP. So this little stick figure representation right here, this is your Z disc. This right here is troponin, tropomyosin, and actin all together. So it's the actin covered by tropomyosin, which has troponin bound. Just because it's easier to draw and I don't want to have to duplicate that a billion times. So then, this is our state at rest, no calcium, nothing extra in the cell. From here, when we have this calcium flow down and we add calcium and it binds, the calcium actually is going to bind to the troponin. It binds to the calcium subunit of troponin. Two calcium molecules bind to the low affinity sites for calcium and troponin, and we have, oh my gosh, seriously, go black. Okay. The actin is exposed, the active sites are exposed, and the myosin binds, still ADP and phosphorus, it's hard to see because I wrote it really small, but the myosin binds to the F-actin. Then energy is released as heat, because when we make a bond, we release energy. And then we also are going to break this bond with phosphorus as we do that, because there's just a little bit of extra energy I'm also going to move all of these over really fast, just so that I have some more space. So then as we go through the cycle, we are going to release this phosphorus, and we're actually also going to release the ADP. And what that's going to do is cause a shift in the myosin to go from cocked, right here, the myosin is cocked, to uncocked. Like I explained earlier, there's two conformations. So it goes from cocked to uncocked. Now it has no ADP, no phosphorus. So it's in a pretty low energy state, uncocked. So in order to get it back to a high energy state, back to being cocked, we have to add ATP. And when we add ATP, we break the bond between, I'm just going to put a T there, it stands for ATP. We break the bond between the myosin and actin because we're adding an ATP. And we also are going to release heat again because we're breaking bonds. Then the last thing that happens, myosin in its head has ATPase. So it has the ability to break down ATP into ADP and phosphorus. So we are going to hydrolyze, is the word for it. We're going to hydrolyze ADP, or ATP, into ADP and phosphorus. And that will cause myosin to go from uncocked to cocked because when you have a phosphorus on the side, there's not enough space for myosin to be in that low energy state because we call it steric hindrance in chemistry. Basically, the phosphorus gets in the way, so the ADP can't be cocked without the phosphorus getting in its way and just really causing some problems. So we call this the cross-bridge cycle. And this is the cycle that myosin goes through as a muscle is contracting. So each sarcomere has a lot of myosin that are each going through their own cross-bridge cycle. They're each at different points in their cross-bridge cycle. And they will, as they all do this all together, the sarcomere will shorten. And that's how we get muscle contraction, as each individual sarcomere shortens because each individual myosin is going from cocked to uncocked, unbinding, and then binding to another F-actin that's further along in the sarcomere. So then, when that happens over and over and over again, the muscle obviously is going to get shorter and shorter and shorter until you're fully contracted. Okay, so we obviously have to use ATP to contract a muscle. So where does the ATP come from? And it actually comes from several places depending on how long you're contracting the muscle for. So the first place that your body goes to for ATP is the cytosol. So we have cytosolic ATP. And that's just ATP that's being stored in the cytosol of the cell. This can last for about 1 to 5 seconds. Sorry. So it lasts for 1 to 5 seconds. It is anaerobic, so it requires no oxygen. And this is good because you don't have to go, your body doesn't have to go anywhere. It doesn't have to make anything. It's just kind of there. But it's bad because it lasts a really short amount of time. So you can get 1-2 jumps out of it. That's about it. A really limited supply, not super great in the long run. The second place your body goes to for ATP is called creatine phosphate. And creatine phosphate is a substance that is stored in your muscle cells. And it takes, using it, we can go from ADP plus, so I'm going to put CP, that's creatine phosphate. We can use a kinase, we call it creatine kinase, to add a phosphate to ADP and make ATP. So this is a little bit easier than going and getting an inorganic phosphate and adding it to ADP. So our body goes to creatine phosphate next because we have a kinase right there. It's really easy to use, and we just pop on a phosphate from an organic source. This lasts for, from like the 5 to 10 second range. It's also anaerobic, and it's good because it's easy. It happens really fast. It's already in our muscles, and it's easy. But it also lasts a really short amount of time. So that's not good if you're trying to run a marathon. Not going to work for you. So the third thing that we can do is glycolysis. And glycolysis is something that people are typically familiar with, but glycolysis is also anaerobic. It's going to last for 1 to 2 minutes. So this one is good if you're, I'm a swimmer, so when I swim my short distance races, I am probably exclusively using glycolysis, especially considering I don't breathe a lot. I really can't use a ton of oxygen because I don't have a ton of oxygen in my body because I usually don't breathe. So unless I'm using oxygen from my beginning state, I'm not really using it at all. And my races are definitely shorter than 2 minutes. So another thing that, could use glycolysis is like running the 100-meter race or the 400-meter race even is probably all glycolysis. And glycolysis is where we take glucose and we break it down into ATP. And this one is good because we get some energy from glucose, but it's not as good as trying to break glucose all the way down using an aerobic method and a method that uses oxygen. I figured I should probably be more clear about that because it sounded like I said anaerobic. But it's not quite as good as a method that uses oxygen because we don't get nearly as much energy at all. Actually, the energy that we get from glycolysis is minuscule compared to the energy we get from cellular respiration, from full cellular respiration. So it's not ideal in the long run. Again, you couldn't run a full marathon using glycolysis, but it's good because we get enough that we could run a sprint race, or people that are lifting could use this for, while they're actually lifting. But the last thing that our cells go to is cellular respiration. And this is good for endurance athletes because they use it all the time. This lasts for hours and it is aerobic. So we do need oxygen. And this is where our body takes glucose and we break it all the way down using the mitochondria into carbon dioxide and water. And it goes through the whole Krebs cycle and everything that you learn in previous classes that we don't care about here apparently. But we could run a marathon with this. We could swim 10 miles with this. We could bike all day with cellular respiration. This source of ATP doesn't really run out. In fact, it doesn't run out. Your muscles will fatigue before this source of ATP runs out. So this one is really good because it doesn't run out. But it is more costly and it does require oxygen. So if you're not, if you don't have oxygen available to you, then that obviously isn't going to work. I think that's about it. Perfect. So, there are 2 main types of smooth muscle. One is single unit, and one is multi-unit. And single unit smooth muscle is not connected by gap junctions, so they kind of stand alone and they're controlled solely by nerve fibers. Examples of this is the erector pili in your skin or your eye muscles. And then multi-unit, they're connected by gap junctions. So if a signal is sent to 1, it can travel down the units of the muscle cell. And examples of these is like peristalsis in the digestive system happens because of multi-unit smooth muscle cells. So it happens in the digestive system, in your urinary system, like your bladder, places like that. Molecular mechanisms of muscle contraction. So there's actually 2 different kinds of muscle contraction in smooth muscle. There's phasic contraction and tonic contraction. I'm going to have to separate those further, but phasic contraction, in phasic contraction, the cross-bridge cycle still happens, but there are a few extra components. So I'm just going to draw the cross-bridge cycle like it would happen in skeletal muscle, and then I'll just add the components. So we have ADP and a phosphate connected. Then we're going to add calcium, and we're going to add an enzyme called MLCK, which is myosin light chain kinase. So what's going to happen is it's going to phosphorylate the myosin light chain. And this is new. This doesn't happen in skeletal muscle, so I'm going to make it blue. And then because we're adding a phosphate and we're using a kinase, we also need an ATP. So we're adding an ATP at this step as well. Then we go to step 2, where we bind because we've added calcium. And we have another phosphate here on the light chain. It's important to note that smooth muscle does not have troponin. So the mechanism for ATP binding actually goes through the MLCK. So the myosin light chain kinase makes the myosin more, or makes the myosin have like a higher affinity for the actin. So they're more likely to bind. So myosin binds, and we have a release of energy again as heat, just like we do, and as breaking of bond. So then we release an inorganic phosphate, and we release an ADP. Then we're in a cocked position, still attached, but we have a phosphate attached. Then in this next step, we have ATP. Being brought in. We still have a phosphate attached. Just like it happens in the normal cross-bridge cycle. Then the last step, we have to bring in another enzyme called MLCP, myosin light chain phosphatase, which is going to take our phosphate off. So we have a phosphate leave, and we have to hydrolyze. So hydrolyze, which then puts us back to where we were at the beginning. This step actually can be bypassed in a phasic contraction, so we can bypass and go straight to step 2. Just like that. So we can go right back to rebinding if we keep the phosphate on. In a tonic contraction, the mechanism is a little bit different because MLCP comes at a little bit of an earlier time. So the first steps are the exact same. We have myosin with an ADP and a phosphate. We add calcium, MLCK, ADP. That's actually supposed to be ATP. We have the second step where myosin has bound. We have a phosphate. We have another phosphate. And this is where it differs, because right here, in a tonic contraction, MLCP enters. And so a phosphate leaves in addition to your other phosphate leaving and the ADP leaving. So then we have I messed that up. Ignore me. No, I'm right. So we have this COX molecule here. Now when we add an ATP, this actually makes it harder for the ATP to come in and bind. So it kind of gets in like a power struggle with this myosin head, and the myosin head doesn't really want to unbind. So this step right here gets really long. It's called the latch state. Because once the ATP does come in and bind and the myosin unbinds, then we can hydrolyze. But before that, it takes a while. So that makes it so this tonic contraction can last a long time. So a contraction can last a long time without using more ATP, which allows for things like sphincters to stay contracted without using a ton of ATP, and they can stay contracted for like a really long time without causing an issue. Okay, comparing and contrasting smooth muscle. Okay, so structurally, smooth muscle and skeletal muscle are pretty similar, but smooth muscle doesn't have troponin, like I mentioned, and the fibers are able to overlap more. So smooth muscle can contract to about 80%, where skeletal muscle can only contract to about 30% of like fiber overlap. So the fibers can overlap a lot tighter in smooth muscle than they can in skeletal muscle. Additionally, functionality-wise, smooth muscle is usually controlled without thinking, involuntarily. That's the word I was thinking of. And skeletal muscle is controlled voluntarily or with reflexes, which are involuntary but done through a different mechanism. And to regulate smooth muscle, we use the autonomic nervous system typically, and skeletal muscle is regulated through either voluntary movement or through reflex movement, which is the spinal cord. Okay, the last part is muscle sensory receptors. I'm just gonna zoom in here and so muscle sensory receptors, there are two main muscle sensory receptors. There's muscle spindles, which control the stretch reflex, and the Golgi tendon organs, which control your withdrawal reflex. So the stretch reflex relies on muscle spindles, which if this is a muscle spindle is like a little part inside the muscle. Like this. And if we blow up the muscle spindle, it would have two different types of fibers in it. It has a chain fiber or a bag fiber. And a chain fiber has nuclei, kind of lined nuclei, lined up on the outside, where a bag fiber, the nuclei are kind of, it's like if you put coins in a bag, they're all tied up in one spot. And these fibers are innervated with what we call gamma motor neurons. And gamma motor neurons are going to help the muscle spindle stay taut when your muscle is contracting. So when we're contracting a muscle, gamma motor neurons gamma motor neurons, they keep the muscle spindle taut. They also have sensory neurons running through them that are running out. The ones in the chain fibers, which are these ones right here, are called static sensory neurons. And the ones in the back fibers are called dynamic sensory neurons. And they're called this because when a person is actually contracting their muscle, or it's actually stretching, when a person is stretching their muscle, so if they go from like a taut, contracted state to they're actually stretching, they're in the motion, that's a dynamic movement, they're moving. So that will be something that's sent to the spinal cord in the brain through a dynamic sensory neuron. If they stop moving and they are just holding the stretch, that will be sent out through a static sensory neuron. And that's important because they're coming from two different places. Another thing that's important is that this muscle is innervated using, just work with me here, an alpha motor neuron. And the alpha motor neuron and the gamma motor neuron, they come into the muscle through the same nerve. So when one fires, so does the other, which is important because as we're contracting the muscle, that's what's going to keep the muscle spindle taut, because as the alpha motor neuron fires, so will the gamma motor neuron, which keeps the muscle spindle nice and tight instead of contracting and instead of becoming loose when the muscle's contracting, because otherwise it would become loose and look silly and not work anymore because it wouldn't be a stretch receptor. Anyways, that's kind of the idea for the anatomy, like really basic anatomy there, but the reason that this works is if we have like I'm just going to recreate Brother Hunt's drawing. It was just absolutely wonderful. So if we have these two muscles on a leg, and we have the spinal cord, which is what controls reflex movement. Hold on, I needed to make that bigger. If we start stretching the quad muscle, so somehow we start stretching the quad muscle right here, this stretch receptor, this right here, I'm pointing at my screen like you can see that, this right here is going to receive, actually this, dynamic sensory neuron is going to receive that input and it's going to send it to the spinal cord. So the sensory neuron is going to send it to the spinal cord and it's going to synapse with a motor neuron. There's actually two motor neurons in the spinal cord. And so this motor neuron right here is going to be our quad motor neuron. And this motor neuron is our hamstring motor neuron. And the reason we have two motor neurons that this reflex sets off is because if we want to flex the quad to keep us from stretching too far, then we need to relax the hamstring. Otherwise, our leg isn't going to move anywhere. So the opposite muscle, we're going to have to relax it. So there's another little tiny neuron in between the motor neuron and a sensory neuron. that is going to communicate with the motor neuron for the hamstring and make it an inhibitory potential, because this motor neuron right here, or this sensory neuron, is only going to send out excitatory action potentials. This one is going to send out an inhibitory action potential to the hamstring neuron. That way, we can flex the quad but relax the hamstring so that our leg can contract, or our quad muscle can contract so that it doesn't stretch too far and potentially hurt us. There's another sensory receptor that does kind of the opposite thing. So a Golgi tendon organ is what we call it. That sits actually on the tendon. So if this is your patella, it would have the sensory neurons would sit onto your patella. And not onto your patella, onto the tendon that connects your hamstring or your quad to your patella. And when that becomes too far stretched, the Golgi tendon organ reflex will take over, and it will tell your muscle to stop contracting or to stop stretching, because you're going to put too much pressure on the tendon, or you're going to have too much pressure on the bone, because we have enough muscle strength that we could pull our tendon off of our bone. So this Golgi tendon organ is there so that we can prevent sorry, my brain just died. So we can prevent over stretching. This receptor is a proprioreceptor. That's what it's called. So it's a proprioreceptor. And it helps to tell us about, like I said, the tension on a muscle or a tendon. So if that's activated during contraction, so if we contract the quad too much, what's going to happen is actually the opposite of what's drawn up here. So what will happen is, I'm going to draw a new picture, but just leave the one on the side there. What will happen is the sensory neuron will come in, and we're contracting the quad too far. So if this is our quad neuron, motor neuron, and this is our hamstring motor neuron, in order to prevent pulling the tendon off the bone or hurting ourselves, we will inhibit the quad. I drew this kind of wrong, but we're just going to go with it. We will inhibit the quad muscle by inhibiting the motor neuron through a secondary neuron there. And we will activate the hamstring neuron, which then will contract our hamstring and bring our quad back to a place where we're not going to hurt ourselves.